Hello students, let's take a look at the muscular system. It makes up about half of the body's mass. The muscle can transfer chemical energy into mechanical energy for movement, contraction of muscles. And we look for those root words myo, mys, and sarco all have to do with muscle. There are three types of muscle tissues, and when we were studying about tissues, you learned about these. There's skeletal muscles, usually attached to bones. We know it's usually under conscious control, unless we're cold, and it is striated. We have smooth muscles. It's found in the walls of most viscera. Our blood vessels are lined with them. Our skin has the erector pili muscle. It is not under our conscious control, and it's not striated. Cardiac muscle is found in the wall of the heart. It's not under conscious control, and it too is also striated, a little less noticeable on a slide than skeletal muscle. The function for muscles generate for locomotion, movement of food, blood, urine, lymph, also maintaining skeletal posture, stabilized joints. That's why you go to physical therapy if you have an injury to a joint. It generates heat during the contraction process. Today was a pretty cool day and I got a little chilled, so involuntary muscle contraction generates a little extra heat for those chilly days. The functional characteristics, it has excitability and inhibitability. It is the ability to receive and respond to a stimulus. Contractibility, it is able to shorten forcefully. Extensibility, it is able to be stretched, extended beyond resting length. It also has elasticity, where it has the ability to resume that resting length after being stretched. Each muscle typically has thousands of muscle fibers. Each muscle is innervated, meaning that it has nerves that help to transmit a stimuli to cause that muscle to contract or not. It is the reason for muscle contraction. Each muscle has an artery and one or two veins. Each muscle also has arterioles, vanuoles, all the way to capillaries, and the capillaries are long and windy, almost slinky looking as they wrap around those muscle fibers. They have attachment. Most span joints at 2 points. Contraction moves the insertion toward the origin. We talked about that in lab. There is sometimes direct attachment, fleshy. The epimysium is fused to the periosteum or the perichondrium of aponeurosis, or to the bone. Okay, there is indirect attachment sometimes, where the muscle is attached to a tendon, or aponeurosis attached to bone or cartilage. Aponeurosis is where you have, it's shown here, that's aponeurosis right here on the top of your head. And it's almost like a sheet-like tendon. It's also called a rectus sheath right here. And it connects one muscle to another. It's a sheet-like tendon. Up here connects the frontalis to the occipitalis. That's on the back of the head, the frontalis here. Aponeurosis. And then the abdominal region, you have the rectus abdominis, is covered up by that fascia of that aponeurosis. And remember, a tendon attaches muscle to bone, ligaments bone to bone, tendon muscle to bone. And that aponeurosis that is like a big sheet of fascia. And you see fascia and tendons when you eat a chicken leg. There's like a long string of gristle in there. That would be fascia. Okay, so we did say that attaching muscle to bone is a tendon, and the tendon is continuous with the periosteum on the bone. It is like part of that periosteum, therefore part of that bone. So if you rip away this muscle, you're going to bring bone with it. Okay, continuous with the tendon is going to be all of these connective tissues. Okay, so the outermost is going to be epimysium, and there's that root word mice in these two words, epi, upon, peri, around. So the epimysium is surrounds the entire muscle. In the first of these three distinct layers of fascia of connective tissue sheets that are continuous with each other and actually continuous of this tendon, so it's part of the tendon. It gives the strength to hold that muscle under great stress to the bone. So the outermost connective tissue, epimysium, several fibers are bundled together. And those fibers form a fascicle. That entire fascicle, there's a fascicle, and surrounding that fascicle is going to be perimysium. Surrounding each muscle cell is endomysium. Endo, when you see that prefix endo, just think of the innermost. Okay, so the smallest structure is going to have the endomysium. So here's that fascicle, and that fascicle is divided up into bundles of fibers, a myofibril and a myofilament. Okay, and each bundle is a myofilament. The myofibrils are those fibers that make up those bundles that are in that myofibril. Okay, so the endomysium will surround each one of those myofibrils, bundles of muscle fibers. The muscle fibers are long, about 30 centimeters, cylindrical, and have multi-nuclei. It's multinucleated, and we talked about that when we looked at those slides and talking about tissues. The reason why it is multinucleated is thought because of syncytium or syncytium. There's a whole lot of embryonic cells that help to form that muscle tissues, and because there are so many cells that are bound together, they keep those the nucleus for each cell, and so that makes it multinucleated. And that is why. From the largest to smallest, the entire structure is a muscle, and the muscle is made up of fascicles, bundles of muscle fibers, and then bundles of muscle fibers form myofibrils, and then thin and the thick and thin filaments. Now we have a model similar to this in the A&P lab. Okay, let's go over the different parts. Now this outermost, this would be covered by endomysium here, but it's like a cell membrane, and that cell membrane is called, there's another root word that has to do with muscles. Here's sarco. Sarcolemma is like the cell membrane. Okay, sarcoplasm on the inside here is like the cell cytoplasm. Okay, the sarcoplasm. Then you have sarcoplasmic reticulum. It's like the endoplasmic reticulum of a muscle cell, but because it's muscle cell, it is sarcoplasmic reticulum. And these yellow lines here form a network of tubes for delivering electrolytes, calcium, neurotransmitters, all kinds of things. It's also that bioelectrical impulse if needed. Endoplasmic reticulum, very similar to sarcoplasmic reticulum, but because this is muscle, this is sarcoplasmic reticulum. The myofibrils are like organelles extending the entire length. Up to 80% of the volume of the muscles is hundreds to thousands of these myofibrils in one muscle. The myofibrils are the contractile elements of the muscle. It consists of a chain of sarcomeres lined end to end. So from this C line to this C line, Z line is a sarcomere, Z to shining Z, and that is only one of many lining up one after the other from end to end for that myofibril. Then the sarcoplasmic reticulum, you have the cisterna. The cisterna of the sarcoplasmic reticulum, a little bit larger tube in this delivery system. And then in the center here, this is called the transverse tubule. So cisterna of sarcoplasmic reticulum, cisterna of sarcoplasmic reticulum, and the T-tubule, and all three together we call that the triad. They help to regulate muscle contraction by delivering calcium and other things that are needed to stimulate that muscle contraction. And the smooth endoplasmic reticulum of the muscle cell is the sarcoplasmic reticulum. Okay, you don't see ribosomes attached to it. It surrounds a myofibril with the terminal cisterna cross channels at the AI junction. We haven't talked about that yet, but it would be at the AI junction. Functions to regulate intercellular calcium levels. How we talked about before how calcium's so important, and one of the things calcium has to be present for muscles to contract. It has to be present for the neurotransmitter, the chemical that has to be present for this muscle to contract. To get everything started, you have to have calcium, and we'll talk about that again. But these delivery tubes will deliver your ions, your electrolytes, but calcium's very, very important. Can't have a contraction without the delivery of calcium. Those myofibrils are constructed mainly of actin and myosin filaments. Again, the triad is the two terminal cisterna with the transverse tubule in the middle, and at the AI junction. Now at the AI junction, this is the I band, and here's the A band, and we'll look at that again in just a minute. Okay, so it brings in nutrients also to the sarcomere so the cell can be thick, and it propagates muscle impulses to every sarcomere, so it plays an important role in contraction. from sarcomeres from one Z line or Z disc to the next Z line. Okay, let's look at a sarcomere. When we were looking at tissues, we looked at skeletal muscle tissues, also cardiac muscle tissue, and we saw striations. Striations are like the stripes of different colors on our slide. They're repeated dark and light bands, and these names come from dark and lightness of colors, and that's band of light. Okay, so the A bands, you have the I bands, the A bands, the H zone, Z line, and M line. You need to know about these in lecture, but it's important also to know about these in lab. Okay, so here is that terminal cisterna and the T-tubule there. Let's go over these parts. This area right here, the zigzag line, is called the Z line, and here's another Z line. From one Z line to the next Z line is called a sarcomere. So from this Z line to this Z line is a sarcomere. From this Z line to this Z line is another sarcomere. So this myofibril is one sarcomere after another, attached to one another. Now, let's go over the different parts, and then we'll talk about each one of these. All right, so the I band, the Z runs through the I. So attached to the Z line or Z disc, some books call it a Z disc because it is disc shaped, is the thin protein filament actin. This is the thick protein filament myosin. So attached to the Z is an actin. There's another actin, protein filament actin, the thin protein filament actin, the thin protein filament actin. Now, the myosin appears to be like floating here. You don't have to remember this, but there is a thin protein line called titin that holds these myosin filaments in place. And the myosin goes from here, the thick protein filament myosin from here to here. Okay, now when we looked at that slide, we saw striations, and this is sort of like what we saw. Imagine that the light is shining up through the opening of the aperture on the stage from your light source, and it is shining through this section of muscle on that slide. Since you only have this Z line and the actin, which is very thin, the light coming through is very light. Okay? It's very light in color. Okay, this is a stained slide, and it's going to be very light in color. Now, here's the myosin filament, protein filament. The light's coming through, and you have actin and myosin overlapping each other. So when the light goes through there, actually not much light goes through there. So this area on that slide would be the dark part, the darkest part of the striation. Now in this area here, we only have myosin, so it's going to be medium dark. And then another dark part of that striation. And then lighter, because this is only actin and then that Z line, and then you repeat for the next sarcomere. So that is why you have the appearance of those striations on that slide. You're actually seeing light penetrate through that very thin section of muscle tissue. So let's name these things here. All right, the I band is between the end of the myosin here, the end of the myosin here and the end of the myosin here. The Z runs through the I. Okay? From here to here is the I band. Now, the A band is the length of the myosin filament. So from here to here is going to be the A band. From one end of the myosin to the other end of the myosin is the A band. So the A band is actually going to have a dark area, medium dark, and then dark again. So here we have the dark, medium dark, because this is just myosin here, but myosin's thick, so it's not as light as the I band, and then dark again. So the A band has dark, medium dark, dark again. So the length of the myosin is going to be the A band. Okay, the H zone is where you have that medium dark. From this end of the actin to this end of the actin, medium dark here. In the middle is where that thin protein tightened filament is, and that is called the H zone. And in the H zone, by the way, is the M line. This darker pink line right here is the M line. That's where that protein tighten would be holding that in place. Okay, this is the M line. In the middle of that A band, there's the H zone. In the middle of the H zone is the M line. There's another Z line. From Z to the next Z line is a sarcomere. Okay, and here's a drawing of that sarcomere again with the different areas. We have the Z line here and the Z line here forming the sarcomere. Attached to the Z line or Z disc is going to be the thin protein filament actin. Actin, actin, actin. And then here's the thick protein filament myosin. These two filaments play an important role in muscle contraction. And remember, this area, this lighter band of color, is called the I band. And in the middle is the Z line, the I band. From this end of the myosin to this end of the myosin is going to be the A band. Where it overlaps is going to be dark striation, medium dark, dark where it overlaps here. In the middle section where the actin and myosin do not overlap is called the H zone. Medium dark where the actin and myosin do not overlap is called the H zone. The middle here holding the myosin in a straight line would be the M line. So thick filament myosin, it's a protein that extends the entire length of the A band. The M line holds the thick myosin filament together. The thin filament actin is attached directly to the Z line, and it goes into the A band and stops right here in the H zone. It is part of the I band and the A band, but it stops right where the H zone begins. They do not overlap. Okay, there are protein subunits on these two protein filaments. The thicker for the myosin, you have these myosin heads. These myosin heads are called actin binding sites. They are flexible and have sort of a hinge effect here, and we'll talk about that more on the next video. The actin, the thin filament, has regulatory proteins bonded onto this thin protein. There's the actin protein filament. And if you will think about this as beads, two strings of beads twisted around, and this string that is holding these beads together That would be the tropomyosin. And then attached to the tropomyosin are going to be these larger beads called troponin. This is a type of protein, the tropomyosin, and this is another type of protein, the troponin. Okay, just to make a quick summary of what happens here, you have the influx of calcium. The calcium attaches to the troponin. The troponin changes the shape of this actin filament, and it will untwist to expose the active site. And the binding sites for the myosin, you have to have calcium for the binding sites to be exposed for the myosin, so that you can have, here we see on this drawing where they're touching. They're not always touching, okay? Only when there's calcium and enough ATP and the electrolytes are right, they'll touch, and you have a stroke of this myosin flexible hinge region, this myosin head, and it actually pulls that actin along. And we'll talk more about that in the next video.